Good morning, fellow researchers, respected session chair. My name is Arunangshu Ghosh. I work as an assistant professor at National Institute of Technology, Patna. This work has been the contribution of undergraduate students, postgraduate students, and it is also a part of a project that is running and funded by Government of India. Development of electronic interface for sensing applications with voltammetric electronic tongue. Since morning, we have been talking about biomimetic instruments. Electronic tongue is a type of a biomimetic instrument. The presentation summary goes like this. We will introduce what is an electronic tongue, some applications. Lots of applications are there. Those will be, few of them will be reviewed, and components. What are the components of electronic tongue? And then we will move on to the electronic tongue development circuits. And we have also conducted few experiments to distinguish among different types of test samples. And we will present the results and analysis and finally the conclusion. So as we can see that in this work, we are going to develop, or we have developed, an electronic interface for voltammetric electronic tongue meant for analysis of liquid samples, mainly targeting the spoilage detection of food samples. Some few liquid samples like black tea, green tea, and of course impure water from tap, and also pure water from millipore water purifier system and milk has been analyzed. And signal processing techniques like discrete wavelet transform has been used for feature extraction. And finally, principal component analysis, this technique has been shown to render, has been used to show that the multivariate response is clearly separate from each other when the samples are changed. Electronic tongue is basically an array of electrochemical sensors. The human tongue is also an array of receptors. The stimulus, whenever some food chemicals interact with the olfactory, gustative receptors, produces some chemical reactions. Those reactions are travel to brain and they're interpreted as a fingerprint. The same philosophy has been applied here. An array of electrodes, electrochemical sensors are used, and when they are presented to a mixture of chemicals, very complicated chemicals, chemical matrix, like typically what comes from a natural source, some oxidation reduction of charged species take place, and the combined response gives the fingerprint about the sample that is being analyzed. And then the data analysis techniques like statistical and as well as artificial intelligence motivated techniques can be used to generate the separation or classification and then quantification task also. There are lots of applications which with lots of literatures are there. Typically, the first electronic tongue was presented in the year 2000, but it was a different type of electronic tongue. The tongue on which I am working, the voltammetric electronic tongue, it was presented, the philosophy was presented in 2003, around 2003. And since then, it has been in a wide, very, very wide use. So the development involves these three things: the electrode array, the electronics part is the potentiostat module, and the software module also. Well, the three electrode system is actually a principle how the electrodes should be arranged. And the working electrode, as we can see, is the one which is used where the actual electrochemical reactions occur, and there are two supporting electrodes, reference electrode and counter electrode. So that makes a three electrode system. But this is not an array. Whenever we are using an array of working electrodes, then it becomes what is known as the electronic tongue-based and array-based signal processing methodology. So there is one more aspect to this particular electronic tongue, is that we are using some probing voltages, pulse voltammetry. It's a rectangular shaped sequence of pulse waveform with time-varying electrochemical potential is applied to the working electrode. The current that flows due to redox reactions occurring due to the components present inside is measured. So obviously we have I to be converted here. We are going to present the circuit, and the role of this device, there is some device known as potentiostat device, is to control the amount of voltage that is applied to the reference electrodes. We actually cannot directly apply the voltages because there are lots of drifts that occur, so we need some specialized circuit. And this potentiostat device not only controls the applied potential to the working electrode, but it also measures the current that is available as a part of response. So here is the electrode array. We are using, there are 5 working electrodes, as we can see. These are the working electrodes, a very crude one, a first prototype that has been built: gold, iridium, palladium, platinum, and rhodium. Those 5 noble metal electrodes are used. This one is the counter electrode, and in the middle we apply what is known as a reference electrode, which we have selected as a silver chloride reference electrode. So basically only one electrode forms the 3 electrode system at a time. For example, gold is a working electrode, stainless steel, and then the reference electrode. This forms a single 3 electrode system. After collecting the response from the gold electrode, the control now switches back to the next electrode. So it's a sort of sequential collection of response. Well, this part has already been discussed. Potentio means voltage and stat means to keep constant. This is an electronic device that maintains a constant voltage between reference and working electrode. Reference electrode is the reference of an electrochemical cell. So this potentiostat is an electrode, well, the basic circuit of controlling the potential, collecting the response, that is same. But along with that, what is being added is the electrode array switching circuit. So that is applied because we basically have an array of working electrodes. So it also houses one data acquisition system which has been developed in-house, and then an in-house developed front-end interface for controlling and sequence control operations and rendering of the response. So this is a software module which has been developed. The software module, the job is to basically initiate the operation, pass on the starting parameters to the microcontroller which is there in the data acquisition system, and then once the responses are acquired, we can plot it. And of course, data logging is supported. So this is a block diagram. We have basically the 3 modules: software module, hardware module, where the data acquisition system and the potentiostat part is present, and we have the physical part, sensor part, the electrode array. So the work which is being discussed will be involving this part and a little bit of this part also. A little bit clarified view of this. We have a PC based interface developed in LabVIEW, and we have a data acquisition system where these are 2 16-bit ADCs and DACs. We need ADCs because we need to collect the digitized response. We need a digital to analog converter because we are applying a probing voltages as a part of voltammetric theory. So this has been applied through the potentiostat module to individual working electrodes through the electrode switches circuit, and basically the potentiostat applies and then collects digitization, and then the response goes back to the LabVIEW software where data logging and data rendering also takes place. So this is a microcontroller that has been used for this work, a low power microcontroller supporting all the basic facilities that an embedded system requires. And we have used basically ADC like AD977 and DAC 714. The motivation to select these ICs is that they have a very wide dynamic range, plus minus 10 volt of input and plus minus 10 volt of output for the digital to analog converters also. The circuit, the operating protocol is quite simple, and these ICs I have found they are pretty good as far as flexibility and simplicity of operation is also when considered. So this is the overall block diagram of the data acquisition system that was developed, and this is the circuit, the first prototype. We are in the process of miniaturizing this circuit also. So the whole data acquisition system supporting the parallel lines for parallel port operation to switch the electrodes and all those things is developed. This central microcontroller. We have the DAQ 714P and AD977 interfaced via the SPI protocol. And this is the sensor signal conditioning circuit. This is the part from where the digital to analog converter produces the voltage, and then it is applied to the electrochemical cell. The interesting part is that the electrochemical cell actually is a part of this feedback loop, where the property of the solution gets embedded inside this control amplifier. And whenever some voltage potential is applied, a profile of voltage is applied, the corresponding chemical reactions, which are dynamic in nature when they occur, is converted into voltage signal through this I2V converter, which is again is a gain selective network. So we are actually trying to support measurement of current from nanoampere up to milliampere level. So we are using 1 megaohm, 100 megaohm, so pretty big resistances right now, buffered output stage, and they again go to the analog to digital converters. So we are using an electrochemical grade operational amplifier AD549. This has a very low input offset current in the order of picoamperes. Our measurement range is in nanoamperes, so it should not create problems. So just now the quick, very quick circuits which are being developed. Please let me tell you, this is a work of my undergraduate students, so the circuits are pretty crude right now. We are in the process of miniaturizing them. But this is the electrode array and the power supply module. One of the reviewer raised a question that why the power supply module is so big. Yes, it is big because we are trying to right now use a linear power supply, and also the 5 volt, plus minus 15 volt, 12 volts, all those different types of power supplies are there. So we didn't decide to go with the chopper mechanism of generating the power supply, because that introduces some high frequency noises. So we decided to go on with the linear power supply. This is the potentiostat circuit, the electrode switcher, and this is the data acquisition system. The instrument has been now put in the cabinet. It is a programmable interface. So this is the interface that has been developed in the LabVIEW. We show the characteristics of a test solution in the form of a shape which is known as a voltammogram. So the applied voltage versus, so the applied response current versus the applied voltage is what we get. And this shape actually tells about the characteristics of a solution. There is a very good amount of electrochemical backing in electrochemical theory regarding these analysis. So in the experiment, we have tried to analyze this 5 types of samples, whether the solutions could be identified or something. Yes, so we have also tried to perform 2 types of analysis techniques using large amplitude pulse voltammetry and cyclic voltammetry. So these are the results. As we can see that different types of electrodes are being probed with a triangular voltage, which is actually a theory according to the theory of cyclic voltammetry, and we obtain a current response. These peaks that we observe here are the characteristics of the samples, and it also gives a fingerprint about the qualities of the sample, which can later be embedded into a pattern recognition engine. The LabVIEW response, we can see some spiking behavior here when we probe the solution with a large amplitude pulse voltammetric signal. So we get some response like this. Now there is also lots of other signal processing techniques. We have used discrete wavelet transform for feature extraction, because typical is a very large waveform. So yes, using DWT, we have compressed this many number of data points, 7,400 for LabVIEW, into 115 features. And then we have performed what is known as a principal component analysis. It's a dimensional reduction technique used for multivariate visualization, but only in 2 or 3 dimensions. So as we can see that different types of T samples could be separated. And an interesting thing is that pure water and tap water, they form such a kind of a distinct cluster. Of course, we have seen that when water was kept for a period of time, it started to show some drift, indicating that impurity is creeping in. So this is the same response using the LabVIEW mechanism. And when we compare them, what we see is that LabVIEW comes as a very superior technique. So it produces a separability index. The separability index is a statistical measure how far the clusters are separated. So there are also lots of other work, but in this work we have only presented this two matrices to indicate that the devices that we have developed, the signal conditioning elements that we have developed, are able to distinguish between different types of samples, and LabVIEW comes out as a good candidate. So this is the conclusion. The PCA plot shows distinction, and further works can be done regarding the development of data analysis techniques and also refinement of the signal conditioning circuits.
Thank you. That's fine. Before I